We're very happy to welcome you all here today. So this is the 4th year that we are presenting this event. My name is Ganesh Ramachandran. I'm a physicist and a faculty member at the Institute of Mathematical Sciences. So this has been our annual tradition for the past 3 years. We have an event where we bring 4 exciting talks about current topics, about research that's happening in India, around us, and most of it in publicly funded institutes and universities. We have 4 such talks today for you today. We have the first speaker coming up, Professor Sandhya Koushika. Before that, may I please ask you to turn off your cell phones or put them on silent mode. Preferably, do put them on silent mode and use Facebook and Twitter to send us questions. You can send us questions for any of the 4 speakers today. After every talk, we'll take a few questions from the stage. So we'll take some of your questions received from SMS, Twitter, and Facebook. I will put them to the speaker on your behalf, and we'll have an answer. At the end of all the 4 talks in the evening, we'll once again have a question and answer session. We'll have as many questions as we possibly can that we'll try to get answered by our speakers. Good. So without much further ado, our first speaker is Professor Sandhya Koushika from TIFR, Mumbai. So she obtained her PhD from Brandeis University. She has then spent time at Washington University, St. Louis, and NCBS Bangalore. She's currently at TIFR, Mumbai. As a Mumbai resident, she's very interested in traffic jams. So you'll hear a lot about traffic jams in her talk today. And she also happens to come from a musical family with members who have performed in the Music Academy before. So this will be her maiden performance, I think. We'll see how that goes. So she's won many awards. Perhaps one is the Howard Hughes Medal. So she'll tell us about traffic rules in neurons, about commonalities between the brain, ants, and traffic. Over to you, please.
Thank you all. I'm understandably a little nervous because, you know, as he said, I've had relatives who, my own mom, who's in the audience, who sang here, and my aunt, and I'm certainly not singing. So I and my lab are deeply interested in the brain and the nervous system in general. And in fact, this has been an interest of mine since I started graduate school. And really, what drives scientists is a curiosity-driven sense of what do you want to find out. So all scientists at some level approach questions where, at least as biologists, we are very interested in understanding how something works. So you can ask general questions. How does emotion work? How do you perceive the color blue? How does any natural phenomena around you work? In our case, or in the case of what we do, we look at how does the nervous system work. And anytime you ask the how question, it's of course predicated by why. Why should you study the nervous system? Typically, when I give a public talk, which is not as large a community, I always ask by saying, why do you care? You, the audience over here, who are here today, why do you care about studying the nervous system? And really central to it is, you know, I get a diversity of answers. Usually the answers are, oh, because I want to understand depression, or I want to understand emotion, right? Which are all we understand as reflections of phenotypes or events that occur in the brain. But really, if you strip everything away, you want to understand the nervous system because of 3 things: perception, thought, and action. What do I mean when I say perception, thought, and action? If I am in TIFR, every morning I can see, every evening I can see sunset over the sea right from my apartment. So I perceive a beautiful event of the sun setting. Or I can perceive pain. Say if you injure yourself in some way, you can feel that pain. If you are, like many of us in this room are, in the morning with your newspaper or your electronic delivery of news, you might want, here the picture is drinking orange juice, and every time I look at this picture I say, oh, coffee would be much better. Who wants to drink orange juice early in the morning, right? So that is thought. And finally, action. My ability to move and point, that's action. I want to show that I can actually, you know, show an individual, a cartoon here with a person facing pain. Likewise, if you want to ride the cycle or walk, or in this case when I even move my hands, these are all the outputs of the nervous system, right? Perception, thought, and action. And of course, things like memory and emotion as well. The second thing, which is equally important, is injury and disease. Many of you in this room would have at least heard of Alzheimer's disease, right? Is there people who haven't? Please come and speak to me afterwards. We know that there are catastrophic illnesses that arise due to problems in the nervous system. Here I'm showing you a picture of someone, if you were alive in the 80s, was unmissable. Christopher Reeves playing Superman. What happened to him was he fell off the horse when he was riding, and his entire skull detached from his spinal cord, right? So he had a huge fracture over there in his neck. So these are severe injuries that he faced, and he was Superman, pretending to fly over the whole world and rescuing things, and had kryptonite, which prevented him from doing anything, ended up in a wheelchair. But in our real everyday lives in India, we know that there are many people who have, for instance, motorcycle injuries. Very, very common, and can end up with catastrophic problems due to injuries to the nervous system. So again, just fundamentally to understand how the nervous system works, but also to understand disease and injury, these are key motivators as to why many of us are very excited to understand and work on the nervous system. So what do neurons, which are cells within the body which underlie the nervous system, actually do? They sense, integrate, and transmit information. So I am standing here on the stage, and this light is coming straight to my eyes, right? So I sense my environment around me. At the same time, if someone feels hungry, feels a sense of pain, that is sensing your internal environment. So the nervous system senses the information that arises outside of your body as well as within your body. Then it integrates that information. Oh, I pricked my finger with a safety pin. That's okay, I can ignore the pain, it'll go away in a few minutes. Or I really did something bad to my ankle, I do need to go see the doctor, right? So you integrate that information that comes from sensing all this information, and then you transmit a certain kind of output. What that output is presumably appropriate for the stimulus that you have sensed. For instance, if you're hungry, go and get something to eat. All right. The underlying nervous system, which does this over here, drawn as blue lines, if you actually magnify it, is completely like bunches of cells, these are all pictures of neurons, which are like wires which are interconnected with each other, right? So many of you would have seen electrical wires which run parallel to each other, and I certainly have done this myself in my lab, but all of us have done it or seen other people do it, where you want connection across things and you have this cut wire and you just twist it together, right? So you're actually making a circuit. This formation of circuits with these connected wires using neurons, which are long cells, very, very important to transmit information from, say, your big toe all the way to your spinal cord or even to your brain, right? So this underlying circuit where you connect neurons is very important for all kinds of action, and many of you would have seen this in textbooks. You have a reflex action, right? You walk, I usually give this as an example. I'll walk into a table, bang my knee, and then withdraw it because I'm in pain. And this sensation of pain because you banged your knee against something, or if you go to the doctor's office and he uses this kind of hammer, goes to your spinal cord through one kind of neuron, you integrate that information, and then you have an escape response where another neuron says, oh, muscle, respond to it and pull it back. So an individual neuron talks to or communicates with either another neuron or another muscle, another cell, which is the muscle. And that's how your entire organism, in this case either a human or any other living animal, responds to the world around it. Okay. Finally, in this wired circuit, the ability of one neuron to talk to another neuron depends on the presence of what are communication hubs. Here's a picture, a real picture of a communication hub filled with lots of materials. And so if I bang my elbow or my knee, the neuron which gets stimulated or senses it releases these chemicals known as neurotransmitters when it talks to another neuron, and this chemical is the language by which one neuron talks to another. These communication hubs, for those of you who are biologists in the audience or have studied some biology, are called synapses. So I talked about these wires of neurons which connect to each other. What is very clear in this description that each individual neuron or wire is going to be important. So if I bang my knee, there is one neuron which is going and saying, oh, you know, you got hurt and you need to respond, right? So each neuron within this circuit needs to know what is happening within it. So one of the reasons people started looking at individual neurons really started with World War II. In World War II, countries in Europe were hell-bent on destroying each other. They had this wonderful invention which they were making, you know, developing a lot of planes, populating them with young men who were in the prime of their lives, which were getting shot. So you had young people falling off and crashing their planes and having catastrophic injuries which did not allow them to either move or had severe brain deficits in terms of memory and higher cognitive function. Once people identified that this was because of neurons in the brain, there was a lot of interest in trying to understand neurons themselves. So there was one scientist known as Paul Weiss in University of Chicago. He, amongst others, but I'm showing you a picture from his work, where he started looking at these wires. And he did a very, very simple experiment. We already knew that there was material that was present in the communication hub. You know that these wires are very long and present, you know, can connect long distances, can connect two different parts of the animal. I did a very simple experiment, what I call the version of the garden hose experiment. And he tied a little knot, didn't crush it completely, but he compressed it, sewed the animal up, and let it go for several weeks. And what he saw was in both sides, so think of it as a garden hose which you're watering, you know, not a very stiff plastic hose, but a, you know, sort of flexible plastic hose. If you have a tap of water and you sort of compress it over here, you'll get a bulge, which tells you that water is flowing from the tap up to the point where you have compressed it. And what you saw was the material, you saw this bulge on both sides. So you know that material was flowing in both directions. So what you learnt from this was these long wires are very, very active. There are things that are moving inside it. Okay. You can do something very simple. In the water hose analogy, if you peel it open, if you peel open the pipe, the water will go everywhere, right? But in neurons, what you can do is just take off the skin here or take off the cell membrane and just peer inside using a microscope and see what happens. Such experiments showed that there were things, this is a very, very, very old video from actually squid, which is a marine animal. You sort of squeeze out what is present in this wire and you see things moving around, jiggling around. I'm going to generically call all of this cargo, that is things that move, right? So you could see that things were moving around. Then you use a higher power microscope and maybe, you know, move it around a little bit more. And what was seen again by people like Ron Vale was these tracks and cargo moving along them. So you can see here And I think all of you can see that the things are moving along these tracks. I'm going to play you a similar movie here. And setting up here my first analogy. Don't you think one of these roads with things moving is very similar to what you see on highways, right? So essentially you see a lot of movement of material taking place along these roads. These are cellular roads and these are human roads, right? When we see this analogy, one very simple question to ask is, are there traffic rules in neurons? For someone who spent significant time in Bangalore, I want to say if there are traffic rules in humans and they are from Bangalore, I would dearly like to know who follows them. Seriously, Chennai is not so bad, and neither is Bombay. I don't know who, Ganesh? Bombay is actually part of Bombay I live, very good with people follow all the rules. Less so when you go out to the suburbs. Okay, so the contrast here which I'm setting up is what happens within neurons where material is moving, and one way to be inspired to think about these two questions is to see what happens on roads that we experience every single day. You know, every person experiences, right? The minute they start, young children start going to school, they are either taking auto rickshaw, bus or car or walking. You experience some sort of movement along roads. So what is set up is the motivation as to why you might want to study some of the questions that we are addressing within the lab. But equally important, and which I said, is what is the question we can answer is predicated on how we do the experiments, right? Because you need to be able to do an experiment to find out accurate information again and again, to come up and understand what are the principles. So, how do we study the process of movement within neurons? One very simple way is what I call, you can take, supposing you were standing somewhere in outer space, maybe you're too far away, come closer, come to atmosphere, but you're in a helicopter, and you want to understand, say, I'm going to label all cars green and all trucks red. And then I can see how trucks move compared to how cars move, or how cycles move. So essentially you're painting your cargo with one particular color, allowing you to track it. So likewise, in animals, and here we are looking at a little soil nematode that's known as C. elegans, we paint one of the cargo with a label. Here, seen in white in this black background. Okay? These places where you see a lot of this cargo are actually the communication hubs. Okay? You can now take such a living animal which is moving around, put it under a high magnification microscope, and now you can see cargo moving along the neuron, much like what I showed you with the prior movie, but this is inside a living animal, not torn up and squished to pieces. You can easily see things are moving around. So this is just the version of saying you've painted the cargo, you can label everything, what do you see? Now the final piece, we now have a way to study how cargo move in neurons. You have to ask the question, what did we learn? Do you now have an ability to understand what the rules of traffic in neuron are? For the first, I will just point you to this movie where we again painted the cargo, trapped the animal, again a living animal and you see cargo move in both directions. Very nice, but lots of them don't move at all. Here's one set, here's another, this moved a little bit and then stopped, right? So what we realized when we were making these types of movies, and anybody who has done these kinds of experiments will see lots of cargo moving, but lots are not moving at all. Usually at this point, I tell a little story about the story behind the science, because for a long time, we did not focus on these cargo which were not moving. And Gautam, who's here, who stole my thunder by showing one of the movies which I'm going to show in the introductory video of IMSC, kept asking us, and he's a physicist and I'm a biologist, as different as you can expect, you keep asking us, why, what is all this stuff which isn't moving at all? I say, oh no, I don't know, everybody sees it, we're not going to worry about it. And after you asked me about the 10th time, I felt great sense of shame. I said, if I'm a scientist, I really should understand the natural phenomena around, around the process that I'm interested in, and started looking at it much more carefully. It turned out to be a really fun thing to do over over several years after that. When you go and look inside the neuron, and I showed you things which are moving. Now let's blow it up even more with greater magnification. Right? A neuron is sort of like Indian roads. I was going to use the analogy of Mandarli market where my parents shop, but I went through something called Jam Bazaar. No different, okay? Today when we were coming here, we went to some place called Jam Bazaar. You have roads and you have all kinds of things which are present on it. These are the little cargo that move, right? So I call these the fruit and flower sellers of Indian roads. You have a nice, beautiful road. Someone will park one cycle, somebody else will park 2 cars, someone will double park something which they shouldn't, and you have all of these fruit and animal sellers. So what happens if you are a vehicle who are trying to move through this? You have great difficulty because the space becomes constrained. Does that what happens in neurons? Because when you look within the neuron, you see the same crowdedness with lots of elements being present. And in fact, our experiments show here is the version, the green is the version of the fruit and vegetable sellers, which are locally present filamentous structures called actin in certain regions. The cargo that move are basically like little cars that are moving, and where you have these fruit and vegetable sellers or actin, your cargo come and stop. Essentially, cargo are not moving when there is no space to move, right? This is occurring in normal, healthy neurons. So what I tell everybody who's worried about traffic on roads, as long as you have roads, traffic jams are an inevitable part of life. They could be long-lived traffic jams where you don't move for long periods of time, or it could be like Jam Bazaar where you just slow down and stop for a little while and then something else will move up and you can go further. No different in neurons. That is, cargo come and halt in regions where the road is crowded or the region around the road is crowded. Okay, so traffic jams in neurons are inevitable due to the design principles of a neuron itself. Where else do you see traffic jams? We discussed humans driving on roads, humans even walking on crowded footpaths, same thing. The other place where you see is ants building nests. And this is not our work. So now you're looking at something much bigger, right? And you see this ant over here is trying to make a nest and make a tunnel. All of these ants have come. There's no place for it to go ahead. They all form a traffic jam, right? So your ability to keep cargo flowing is very, very simply depends on whether you have the space to move forward or not. It does not matter if you're a nanometer scale vesicle, you're a human being in a car or a cycle. Cycle might have an easier time, car will have a less easy time, truck will have the most difficult time. Or you're an ant, which is, probably a centimeter or more long. So the fundamental principle is that if you want to move forward, your path needs to be clear. What type of crowding occurs is, of course, context specific. In the roads in Jam Bazaar, it's the fruit and vegetable sellers and flower sellers. In neurons, it's filamentous proteins which occupy the space. And here, it is the diameter of the tunnel itself. Let me repeat this again. There is one unbreakable rule, independent of the size of the cargo. If there is no space, you cannot move ahead. And this is intuitively something all of us understand, right? Because we see it every day in our life. Then the next question is, if you know that there is no space and you know you're coming up to a traffic jam, what will you do? I know what I would do now, thanks to Google Maps. I'll open Google Maps and say, oh, this is too much traffic, I'll leave after some time. Now, Cell is not able to sort that out. And for all the people who don't use Google Maps routinely and they feel, oh, no, we have to start regardless of what happens, that is an option which you cannot use. But what are the options that you can use, right? The first is, again, think about what would happen if you come up to a traffic jam. You can wait it out. The second thing you can do is reverse and go back. Maybe try another road, but certainly go back from that crowded situation. And here again, the green pieces that you're going to see are what I call these actin filaments, which occupy the space. The analogy is the fruit and vegetable sellers on Indian roads. And I'm going to have to wait for this whole movie to loop, I apologize. Here's looking at a red vesicle or cargo, and you can see it comes up against it, comes up against a crowded region, comes a little further down, and it'll start reversing again. Reversing, hits that, goes back, goes forward, goes back, goes forward. It's really, really trying to get past the traffic jam that it sees in neurons. Okay, so that's one strategy. What about the ants example I gave, which is different from neurons? Do they use the same strategy? And the answer was indeed yes. Here is the same movie, a little bit later in time. It's all crowded up. The ants recognize that it's crowded, and they all start backing out. What are they waiting for? We can see actually the tunnel is very narrow here and is a little wider there. So they work to widen this part. So now you have slightly larger area, but they essentially, it's a looping movie, so you'll see that they essentially begin to move out of rich region, right? So essentially they are going back. So if you see some kind of large immovable object, or you've sort of aligned yourself improperly if you're driving on a road, you will go back and try to see whether that area clears up, or you can try another approach. And the second approach is one which is done as a simulation with Gautam. And what you see is that all cargo will come. And if you don't do anything, nothing will move. No? Eventually everything will stop if you don't take any decisions. But you could actually choose locally to change your track. So if you have a multi-lane highway or multi-lane, there are more than one lane. Let's not look at Jambasa. Let's look at the roads which, parallel marine, the marina. You have more than one lane, and you can say, oh, this one is getting crowded. Let me try another lane. And that's what we think. These are strategies that vesicles, which are small, probably use within neurons, which are very similar to strategies we intuitively as drivers on roads follow or ants follow. Okay. Finally, you can look at DNA, which is a long stretch. It's very small. The present is and you can see here, which is an individual protein. These red spots are places where you have a lot of protein present along the DNA. So it's like a crowded location, right? So I'm now looking at something which is much smaller than a vesicle. And what do these proteins do? Let's just watch. It just hops over this place where it is crowded. And here is an illustration of that. Okay. So coming back, what are the strategies that when you have tracks, and the tracks here you're talking about is either the tracks which are called microtubules along which cargo moves in neurons, DNA along which protein diffuse, ants in a tunnel, or human drivers. These are strategies that all of these situations that you can use to not be held up completely by traffic jams. One, you reverse and go back from that location, maybe try at a different time, which is what happens in neurons, and I showed you that one vesicle which kept trying, or try to change lane and go on a different lane. It can be done in combination with the reversal or independently. And the final is the example which I showed you with the DNA, that you can hop across a little crowded region. Okay? And if you really want to think about that, please watch the Star Wars movies where you see all kinds of flying objects and sort of, there are no roads there, right? There are no roads in air. So you can sort of decide to hop over another flying object which is there. And frankly, I'm waiting for the day we have flying cars. I'll be first in line to buy one of them. It doesn't matter if it costs me all my money. All right. So I'm finally going to loop back. So I showed you some of the principles by which vesicles or cargo in neurons get from one place to another. I didn't make a very strong case as to why this movement was necessary, only that it was present. So let's loop back to the first question of why. Okay. I shall describe two contexts, the context of disease and the context of injury. And I will show you two pieces of information which I hope will make you interested in it. Here are actually differentiated human neurons. Okay, so we're not looking at little C. elegans neurons here. Looking at human neurons, here's a young animal, young human neuron. Cargo is moving nicely. Older one is moving nicely, but it's a little fewer. Here is a neuron which has some kind of neurodegenerative disease associated with it. And what you can see is the young animal, there's some moving around, maybe not as many particles as the young healthy neuron, but in the older animal, older human animal, I shouldn't say animal, in older human neurons, nothing is moving at all. So essentially what you're seeing is that these cargo just accumulate. So if you have a lot of cars accumulating or a lot of trucks accumulating, then a moving truck which comes over here will have a very low chance of moving forward again, right? So this type of slowing down and stopping of transport or stopping of movement and increased traffic jam is a hallmark of many neurodegenerative diseases, for instance, Alzheimer's. Okay. But the second and equally important is when, and here I can show you an analogous experiment that was done by me several years ago. Here is a middle-aged worm moving around fairly well. Here's a middle-aged worm which cannot move cargo very well. And you can see it's not moving around at all well like the normal animals are. So you know that this kind of movement within neurons is also extremely important for the movement of the animal itself. Right? And we're talking here about middle-aged and not old animals. In this case, these animals actually end up dying early as well. The other context is neuronal injury. Here's a little cartoon done by a student in TIFR where we can actually cut the neuron in a controlled manner. Right? So we just cut the neuron. This is neuron, we've cut it, and we watch how it regrows and how you can restore function. And an experiment that my student did, we actually saw that you can restore function in this nervous system, provided when you grow back and reattach, you restore transport. So it tells you that the process of flow of cargo and traffic jams are both crucial in two different contexts, in both neurodegenerative disease as well as regrowth and after neuron injury, if you want to have functional recovery. So I've come to an end. I've used 31 minutes so far, or 32 minutes. What I would like you to take home, traffic jams are inevitable. That is the one unbreakable rule in cargo transport. If you don't have space, you cannot go forward. The way in a track situation where you have either microtubule tracks in neurons, roads, tunnels for ants, or DNA tracks for proteins, there are three ways in which you can not get stuck all the time in traffic jams. One is to reverse and try again. Second is to change lanes. And third is to do a little hop. All three strategies are very useful. Maybe all of these strategies will not be necessary if you're in a trackless world, which you would be in air. And movement is essential. Traffic jams are normal in healthy neurons, and movement is essential to maintain healthy neurons and for recovery from injury. With that, I come to an end. There's the logo of my lab showing here traffic jams and all the things we move. And here's a little C. elegans holding a microscope, and that's our lab logo. Thank you.
Yeah. Thank you very much for that wonderful talk. I'll ask the next speaker to come up on stage while we talk about some of the questions. We've got lots of very nice questions. Please do keep sending us questions. I'll flash the details about the questions in a bit. So we'll put some of the questions from you to the speaker. So we've had many interesting questions. The first is perhaps a little, well, not so surprising. Why is C. elegans so popular among biologists?
The reason C. elegans is very popular is because I think, as a person who studies the nervous system, it has very few neurons, but it has a complex repertoire of behavior. It runs away from things that it doesn't like. It runs towards things that it likes. It has preferences in food. It has preferences in mates, which are all hallmarks of any developing species. In addition, it has great genetics. You should see that if you want to understand something, you have to be able to manipulate the process. So two things are necessary. If you have genetics, then you can manipulate the genes. But what I showed you today is a lot of movies where you can actually see what's happening inside a living organism, which is really very powerful because you're not asking someone to guess. You can actually see what's happening. These are the reasons why C. elegans is so popular.
Quite a personality. But I had a related question. How do you see the neurons in human beings?
Okay, so those are actually stem cells from patients which are differentiated in vitro. So using stem cell technology, you can differentiate and make them form neurons in vitro and then actually look at what's happening. Very, very powerful technology again.
No humans were harmed in
No
these
humans were
experiments.
harmed in those experiments.
Good, thank you. So here are a couple of more interesting questions. Here's a question from Gururag Kalanidhi. The previous question was from Shubha. The Gururag Kalanidhi asks, does the movement of cargo through neurons correlate with cognitive processes?
So, in fact, one of the things I didn't talk about, and I said that the traffic jam control can be local, which is the way when we drive, it's local. But it also could have global control. So what has been shown is that when you have situations where you have a lot of transport, and this is in a mouse, you actually see the ability for the mouse to respond to its cues is better. So it does better. It acts like an enriched environment. What we ourselves found in our experiments is that if you sort of stimulate the neuron repeatedly, then something globally happens all across the neuron, in that there are far fewer number of vehicles which get stuck in traffic jams. So we don't quite understand how this global control is there, but clearly there's a global control, and the consequence of that is going to be how responsive your nervous system is, which is, sort of related to cognitive function.
Perhaps a related question from Anita Valliyappan, who asks, what is the cargo actually made of?
So here I talked about specifically, the movies I showed you were all membranous cargo. So they have a membrane associated with them and contents inside it. But cargos can also be just protein clusters. They can also be phase-separated granules where you have RNA and protein present together, but they sort of phase-separate from its surrounding. All of these can move in neurons.
Good. We'll take one more question. We'll have some more. We have time for more questions at the end of all the talks, so please do keep sending us questions, even about this talk. So here's a question from a phone number ending in 4385. There's a common belief that even in a very intellectually bright person, only 10% of the brain is utilized. Does the same hold for neurons? The rest is stuck in a traffic jam.
Traffic jams are present in healthy neurons, and perfectly normal, just like for us on roads. This is a very difficult question to answer because it's very difficult to estimate. But one can say, and I'll say something which is really fun fact which is out there, and we don't understand it. When you now can measure electrical activity, you can take a worm, it's harder to look at it, a human brain. You can look at whole animal. You see a lot of neurons are actually firing and responding and doing things. But when we do other experiments and say, I'm going to remove this cell or this particular neuron, and we know these neurons are important for function, we say, oh, only 3 neurons are important for this function, but when I actually look at the animal, tens of neurons are firing and responding. We don't understand. So obviously there's a lot more activity in the brain than we understand. How do you estimate that you're using only 10% or if it is even the right 10%? I think the answers are all to be discovered. And I couldn't encourage the young people in the audience. Studying the nervous system. One of the coolest things that's going around. And I think anybody will say that. And there's so many unexplained questions here. And I couldn't, if I can even convince a few of you to be dilettantes in trying to understand the nervous system, please welcome.
Thank you very much. On that note, let's thank our speaker once again. Thank you very much for a very nice talk.